Hello guys, welcome back to the Gauravyadavchannel channel. I hope you're doing well. So my today's topic is culture media. So if you didn't subscribe my channel, so please do subscribe it first and watch my other videos and like my video. So let's begin our lecture. So what is culture media? Culture media or the growth medium is a solid, liquid, and semi-solid designed to support the growth of population of microorganism or cell via the process of cell proliferation. So this occur at the optimum temperature and at the appropriate condition. So there are different type of media are used for the growing for the bacteria and different type of cells. So bacteria and fungi have to be grown for them to be identified, and by appropriate procedure they have to be grown separately on the culture media and obtained as a pure for the study. So the history is the original media was used by Louis Pasteur, that was urine or meat broth. The liquid media used in which growth occur as a diffuse growth, and on the solid medium the growth occur as a discrete form. So what is colony? Colony is macroscopically visible collection of million of bacteria or fungi originating from a single bacterial or fungal cell. So the first medium, the first earliest solid medium was cooked cut potato, which was developed by the Robert Koch. So what is agar? Agar is a solidifying agent. So agar was discovered by the Fanny Haze. Fanny Haze was used to make jelly in her kitchen, and the Fanny Haze was the wife of Walter Haze, and Walter Haze was the one of the student or the researcher who was working in the Louis Pasteur laboratory. So from where we obtained agar. So agar is obtained from the seaweeds, red seaweeds, or known as red algae. The species Rhodophycophyta and Giladium pacificum and Giladium amansi. So agar has no nutritive value, so it can't be utilized by the microorganism, but there are some microorganisms which can utilize agar, even like Pseudomonas and Vibrio cholera species. So and there are some advantage of using agar, like it melt at the 90 degrees Celsius and sets at the 42 degrees Celsius, and it remains in the solid form at the room temperature and even in the incubator at the 37 degrees Celsius. So we use 1.5 to 2% of agar for making solid medium. If we want to make semi-solid medium, then we have to reduce the agar concentration, and that will go down to the 0.5%. So there are different type of medium. On the basis of their consistency, they can be classified into solid medium, liquid medium, semi-solid medium, and on the basis of constituent or ingredient, they can be classified into simple medium, complex medium, synthetic medium, or defined medium, or special medium. Special media are used for the special purposes or special type of microorganism. They are enriched media, enrichment media, selective media, indicator media, differential media, sugar media, transport medium, and medium for the biochemical reactions. On the basis of third classification is on the basis of oxygen requirement. So there is a aerobic medium and anaerobic. In today's lecture we are gonna study only aerobic medium. So what is solid medium? Solid medium which has a 2% of agar concentration. So The advantage of solid medium is we can see the colony morphology, where we can see pigmentation, hemolysis, and example of the solid media, neutron agar and the blood agar. On the liquid medium, there is in the liquid medium there is a no agar. So liquid medium used for the inoculum preparation, blood culture, and for the isolation of pathogen from a mixture. An example is neutron broth. Semi-solid medium has 0.5% of agar, and semi-solid medium used for the motility for the motility of the organism, to check the motility of the organism. This is the solid media, this is the liquid medium, and this is the image of the semi-solid medium. This is the picture of growth motility of the bacteria in the motility agar or indole agar. So what is simple media? Simple media, example of simple media is neutron broth or neutron agar. So in simple word, the simple media which has a peptone, meat extract, and NaCl, which has a simple component, is known as a simple media. And simple media used for the non-fastidious organism, means which organism which do not have any special requirement for the growth, such as E. coli. So second is complex media. Complex media, they have added ingredient into the basal media. So why do we add other ingredients in the basal media? To provide the special nutrient for the special microorganism which do have some special requirements for their growth. Third is synthetic media or defined medium. So it is a media prepared from the pure chemical substances, and its exact composition is known only and it is only utilized by some microbes only. Example is peptone water, in which 1% is peptone and 0.5% NaCl is mixed into the water, distilled water. Fourth is enriched media. Enriched media is used to grow the fastidious organism which have some special requirement for their growth, such as Staphylococcus aureus or Streptococcus pyogenes or Haemophilus influenza, etc. So the enriched media is allowed to grow required microorganism without inhibiting the unwanted or non-required microorganism. This is, you can say, advantage or non-advantage, both you can say. So how do we prepare enriched media? What we do, we add blood, serum or egg into the basal medium to make it a enriched media. An example of enriched media are blood agar and chocolate agar. So this is the picture of blood agar, and this is the picture of chocolate agar, and both the media prepared from the blood agar. In blood agar, the red blood cell do not lyse, and in chocolate agar, and we prepare it by the lysis of the red blood cell. Then the cell red, and then the color turn into the chocolate color. What is enrichment medium? Enrichment medium is a liquid media which is used to isolate the pathogen from the mixed culture, and media is incorporated with the inhibitory substances which suppress the unwanted organism. Means in this, first of all, it is present in the liquid form. So if you want to differentiate between enriched or enrichment medium, you can say the enriched media is used to grow wanted and it is used to grow required microorganism without inhibiting the unwanted microorganism, and it is in the liquid solid form mostly. For enrichment media, it is used to grow required microorganism with inhibiting the unwanted microorganism. So it is in the liquid form. And the example of enrichment media are salinite type broth, which is used for the salmonella and shigella. This is the picture of the salinite type broth, and salinite type broth is used for the mainly for the fecal specimen, in which salinite will suppress the coliform microorganism which is usually present in the fecal specimen. And what we get after the inhibition of the coliform, we will get the salmonella or shigella species if they are present in the specimen. And second is alkaline peptone water, which is used for the Vibrio cholera species. So next is selective media. Selective media is the word itself describing itself is selective, means which is used to grow the selective microorganism only. And so the selective media contain inhibitory substances which will inhibit the unwanted microorganism and promote the growth of the wanted microorganism only. For an example, McConkey medium, which is used for the gram-negative bacteria, TCBS for the Vibrio cholera, Lawrence and Johnson medium for Mycobacterium tuberculosis, Wilson and Blaine medium for the Staphylococcus, Salmonella typhi, and potassium tellurite medium for the Diphtheria bacilli. This is the picture of the McConkey agar, and this is the picture of the TCBS agar. And this is also example of selective medium. This is the this one is potassium tellurite medium, and this one is LJ medium, which is used for the growth of Mycobacterium tuberculosis. Indicator medium are media which contain some indicators and these indicators change their color when bacterium grow in them. So this is the first, and this is the image of blood agar containing different type of microorganisms. First is Streptococcus pyogenes, which is showing a beta hemolysis in the blood agar. Beta hemolysis means there is a complete lysis of the red blood cell. This is the image of the Streptococcus colony, which showing the alpha hemolysis, means there is a partial hemolysis around the colony, means there is a green color formation around the colony. So the green color formation is due to the methemoglobin formation, means incomplete hemolysis by the organism. This is third picture is of Streptococcus faecalis, which is showing the gamma hemolysis, means there is a no hemolysis around the colony, means there is a no lysis of the red blood cell. Second medium is McConkey medium. McConkey medium has lactose sugar. So when bacterium like E. coli start growing in the McConkey agar, it will ferment the sugar and produce acid, and it will produce acid. Acid will turn the neutral light indicator into the red dye, and the colony which are lactose fermenter will form pink color, and which are non-lactose fermenter, they will form pale color. So third one is Christiansen urease medium. Christiansen urease medium for use for the urease activity, to check the urease activity, like for the Proteus mirabilis. Next is differential media. Differential media, a media which has substances incorporated in it, enabling it to distinguish between the bacteria. For an example, McConkey medium. As I told you previously, the McConkey medium has lactose and neutral light indicator. So when bacterium start growing, it will ferment. If the bacteria are lactose fermenter, it will ferment the lactose. It will utilize the lactose and produce acid and CO2. Acid, and acid will turn the neutralized indicator into the pink color. If the organism in lactose fermenter, they will form pink color colonies like this, and if the organism in non-lactose fermenter, they will form colorless or pale color colony like this. So next is sugar medium. Sugar medium, media containing any fermentable substances. For an example, glucose, ribose, lactose, starch, etc. They all are carbohydrate source for the microorganism. So media consists 1% of sugar in peptone water and contain a small Durham's tube in inverted position like this. If the bacterium will ferment the sugar and it will change the color. So how it can change color? We will add indicator like a red color indicator, like in this. The first tube is showing positive result because it will turn into the yellow color after the fermentation, and second one is negative because it stays as it is. So if bacterium produce gas, the Durham tube will start floating in the upper supernatant of the medium. So we can check the both thing by the sugar medium for like acid production and the as well as the gas production also. Next is transport medium. Transport medium is used for the transporting the sample, and there are some delicate microorganisms which cannot survive in the time taken process for the use for the transporting the specimen without the transporting medium, like Neisseria gonorrhea. For Neisseria gonorrhea, we use Stewart medium, and which is a non-nutrient soft agar gel containing reducing agent, and second is buffer glycerol saline, which is for the enteric bacilli. This is the picture of the transport medium. And third medium is Carry-Blair medium. Thank you for watching my video, and please do subscribe my channel and watch my other videos also, and please do subscribe my channel. And this is the first part of the culture medium. Second part will also come, which contain anaerobic culture media and the other component of the culture media. Thank you.